My name is Dr. Ben Newman, and I study coronaviruses. All right, let's get our last question for today. It is from Dina, and I once again do hope I'm pronouncing that somewhat okay. Let's see. Hi, Dr. Ben. How can the new strains of the virus be more transmissible? Great question. Yeah, fine. I thought our original version was already very contagious. It was, yeah. Is the new variant here in the US, then why are our numbers of infections going down? Fine, let's get into it. Yeah. All right. Yeah, so how can it be more contagious? Well, a little adage in biology is that any system or reaction can be optimized. So, what we had at the start was a bat virus, essentially, that happened to be pretty good at getting into humans and growing in humans. Now, there are a lot of components that we may not think about that would make a virus a little bit more transmissible. So, if you've got a virus, and let's say you got a virus that can basically get into cells, which is what we started with, that's fine. So, question is, can it bind cells more tightly? And all of these proteins are kind of loose. You've got sort of a shape kind of like this for the spike, and it sort of locks on to a little piece from the ACE2 receptor, about like that. And there actually there's room. You could get a little tighter, a little closer. You could wrap around a little more. Yeah, that's totally possible. But that's not the only way you can get better. So the spike has to be activated. You can make a spike that is easier to activate. Sometimes, if it gets activated too soon, spikes fall apart. So it may well be that in a particular situation, with the way it's being transmitted, if a spike is getting activated too early, then a spike that's harder to activate could be the beneficial one. We see changes happening all over the spike, and some of these changes seem to make generally more transmissible viruses. So we've got changes down near the bottom of the spike that might change how the spike actually fuses with the cell, how it sort of merges the virus with your cell to start the infection. We've got changes that seem to lead to an increase in the production of spike. So spike is it's called a glycoprotein, which means you make a protein, which is like a big long string of amino acids that folds up into a particular shape. Yeah, that's what protein is. And then on the outside of it, I forget how many spots there are, but it's something like 20 or 30. It's just all these spots where the cell has to attach sugar molecules. And there was a wonderful study on this. Each one of the positions where we put a sugar molecule is known, and we're going to try to put the same one on each time that a spike is made. Just to make one brand new virus, you're going to need a few hundred spikes, probably in the order of about 250. That would be a pretty normal amount. And so, if there's a way to make that process faster, and each of these sugar molecules, which are coating the outside of the spike, this kind of act like a camouflage net. Each of those is added and trimmed and branched in a characteristic way. So they each have to interact with a whole bunch of different enzymes that'll just doctor it up just right. Think of the virus coming in for its big interview and sitting down in the chair, and then just a whole team of makeup artists are going, hair, eyes, nose, whatever, and getting everything just perfect before the lights come on. Yeah. And improving the efficiency of any one of those processes can help. You can change the amount of spike that's made, just the bulk amount that you run through there. You can change the way that the immune system is able to chop the spike into little pieces, which is the first part of how it learns to recognize a piece of the spike. And basically, if you can't chop the spike up, then you can't learn from that part of the spike in a particular way. Yeah. And also, of course, the spike protein inside the cell is not just going to sit there and not touch anything else. It's going to interact with maybe dozens, maybe hundreds of different host proteins. And host proteins are like proteins from the cell that have to take it from one place to another. It moves around quite a bit inside the cell as it undergoes its maturation process. Then it has to interact with all the other virus components. And so you can make it a little faster or better at interacting with the virus components, because each of those is also doing the same things, interacting with lots of parts of the cell. And basically, when you're moving from a bat, which is just kind of the same thing, but every part is a little bit different, yeah, into a human, then there are going to be a lot of little tweaks, little nips and tucks that could be made that could make this virus so much better adapted to growing in humans. I think the fact that it's changing shows us, one, that it's kind of flexible, and two, that it's not some bioengineered super weapon that was perfect and crystalline, right from the beginning. Yeah, the virus is changing because changes are making it better at being a virus, and that is what viruses do. Yeah, they're very competitive little things. So there you go. That's the answer to that half. Let's see. If the new variant is here, and yeah, a bunch of new variants here, then why are our numbers of infections going down? Couple reasons. One was the freeze, like if we're just talking Texas, yeah, which I guess I can get off the Texas horse at some point. There's a little announcement on their site that says that numbers right now are artificially low because we weren't able to do testing during most of the freeze. And then a lot of places that were even able to do testing there or after are having trouble getting all their data in and reported. So at some point there'll have to be a correction as they get all these tests actually put into the database, and then it'll look like a giant spike, which won't be any closer to reality. It'll just be all the cases that should have been earlier but concentrated on one day when we happen to get them all through. The other thing is that you've got a lot of different numbers from a lot of different places, and so it's like looking around you and saying, well, it's rainy here today, which it is today. Yeah, I don't see how it could be sunny anywhere else. And it's like, well, no, it's a big planet, big world, big country, whatever. Yeah. A lot of different stuff going on. And so there are places in general where there are stricter following of mask rules and stricter restrictions than wherever you may be. And there are places, one of those would be East Texas, bless them, yeah, where, yeah, the restrictions are much lower, and even the ones that are in place are seldom followed, I would say. And all of this feeds into that same number. If you're looking at a number for a state, or a number for a country, a number for, a continent, region, whatever. So the other part of that is that, thank goodness, Christmas comes but once a year. So we had, in America, Thanksgiving going right into Christmas, going right into New Year. And I think other places we had maybe Christmas and New Year, or some sort of celebration around the same time that would bring a lot of different people together in high concentration from different places, and then they would all go back home. That is how you spread viruses, very much so, and it's always been the case. Christmas has never been a good idea from an epidemiological point of view. It is a thing we do for other reasons which are not connected to science, because we like it. We like family, whatever, which is normally fine, but the fact that we've got a little period here where there isn't a global or big national holiday that would lead people to do exactly the sort of things that lead to infection, that I think is what is causing the numbers to drop. The virus will die out unless we actively spread it from one person to another, and it's got to go right in through the mouth or nose from one sick person to a healthy person who has not been vaccinated or whose vaccine didn't really take. Yeah. And as we vaccinate more, the numbers are going to start to come down. And remember also, when vaccinating, you're taking out, let's say you get somebody vaccinated and somebody coughed on them and they didn't get sick. It's not that you're just taking one little item of data off this giant, curve of infection. You're actually taking off that person, but then that person would have gone on and infected more people, and then those people would have gone and infected more people and more people and more people. And so you're actually, with each vaccination, you're taking something like a wedge, I think, is the best way to think of it, out of the curve. And so we've only done, depending where you're writing from, a relatively small, oh no, you're writing from the US, yeah. So we've only done a relatively small number of first vaccinations and second vaccinations, but that's part of why they're going to have a bigger effect than it seems like they ought to have. It's essentially just mathematics of how viruses grow, and there are wonderful epidemiologists who can explain it a whole lot better than I can. Um, but that, and I think maybe even just the idea of having hope that we can actually get this thing under control for a change, maybe changing a little bit of behavior. So for the ones that are doing what they can and getting vaccinated, yeah, I think they're directly helping, and I think they are making it easier for the people that are not following any of those regulations to not become infected by just reducing the total amount of virus that's out there. So yeah, that's the best explanation that I can find or have heard so far. But yeah, if new stuff comes up, I'll bring it to you, and we will talk about it here. So for now, thanks very much. Very good question, and this has been Ask Dr. Ben.